I know the audience is quite diverse for this particular talk, so I'm going to try and keep it as non-terminology intense as possible. But if there are times when I lose myself and go into ranting professor mode, please do take note and then ask me or clarify any questions you might have later on. All right. So what am I going to talk about today? Well, let me just give you a quick outline. I'll start out by giving you a background on why I'm doing what I'm doing. And since many of you might not be familiar with my field of research, which is evolutionary biology, hopefully I set the stage for you for what I hope is going to be an interesting talk and something different, something that you have not maybe encountered before. And so then I'll dwell into the meat of today's talk, which is specifically about how useful it is to incorporate molecular or genetic information in ecological and evolutionary studies. So specifically, I will highlight two ongoing research programs in the lab, one of which is looking at cryptic speciation. I'll talk more about it in detail later on, as well as a co-evolutionary interaction between dung beetles and the endosymbionts that live within them. And then finally, I hope to wrap up by giving you an insight into why we study insect sex at the Repro Lab in NUS. All right, so understanding the ecological processes and evolutionary forces that drive speciation has been a long-term interest in evolutionary biology. And speciation basically involves the establishment of barriers to gene flow. So for those who are not familiar, imagine you have a species that have multiple populations that regularly interbreed and have been exchanging genetic information for a long period of time. And then suddenly you have a barrier that evolves. Now, this barrier can be a geographical, physical barrier, such as a mountain range, a river, or even man-made infrastructure. Now, these barriers can then result in what used to be a single population exchanging genes becoming two separate lineages that no longer exchange genes. And so you end up having diversification or increase in biodiversity through the accumulation and maintenance of genetic variation among these different lineages. So such barriers over here, as I mentioned, could be physical barriers, but they could also be ecological barriers, such as niche specialization, differences in diet, differences in microhabitat, differences in temporal or seasonal variation. And what my lab is particularly interested in is barriers that arise due to reproduction. For instance, you end up having one lineage that defends a different territory for reproduction or decides to court in a different way. Or you can have another population that differs in its sperm composition or ejaculate protein distribution. So there are many examples whereby you can have speciation via these selective processes. One of the most famous examples of speciation by natural selection can be witnessed across the Galapagos finches, where you have several factors such as separation across islands, local climatic variations, diet specializations, all that have led to species diversification. And there are also instances whereby you have sexual selection playing a role in diversification. For instance, in the cichlids in Africa, where you have non-random mate choice for pigmentation or male coloration, as well as male courtship. So this is the process which we call sexual selection. So for those that are not familiar with it, unlike natural selection, which is often termed as a struggle for existence with respect to environmental challenges, such as foraging or surviving predation or prey, sexual selection is defined as a struggle between individuals of one sex to gain access to individuals of the other sex. And so Darwin himself suggested that the differences in traits that one might observe between the sexes are likely due to sexual selection. So if you find a population of individuals where males and females look different, behave different, now that is most likely due to sexual selection. And as within sexual selection, you can have different types. You can have competition within individuals of the same sex to gain access to individuals of the other sex. And so this is known as, let's say, intrasexual selection, where let's say they defend a territory, a reproductive territory. Now you can also have intersexual selection, whereby there is non-random mate choice by one sex based on mate quality, where you can have one sex being a bit more choosy and preferring mates of a certain phenotype that either survive better or more attractive or more resistant to parasites and so on. So sexual selection in particular can be very instrumental in generating those barriers to gene flow that we talked earlier on. And it can be through a selection of a whole bunch of traits that can be chemical differences, behavioral differences, morphological differences, and they can all be mediated by sex-specific selection, meaning selection acting on males as well as females. And so as evolutionary biologists studying reproduction, we actually know that reproductive traits actually diverge very fast. And comparative studies of numerous species show that the genes involved in sexual reproduction are often more divergent than those in non-reproductive tissues. For instance, the proteins that are involved in basic sperm-egg recognition are some of the most rapidly evolving proteins, and they are not that conserved at all. We can also have rapid evolution in traits that are involved in mate acquisition. So trying to get laid, pretty much. So from species-specific morphological variation to diverse courtship, there's an incredible diversity of traits across taxa. And they can differ even among populations and mediate barriers to gene flow. So to date, there have actually been numerous studies, and both comparative and experimental studies, that have showed the importance of sexual selection in speciation. But one relatively understudied component is what happens after mating. So that's kind of where a lot of the research that is currently occurring with respect to traits that have to do with, let's say, genital morphology or ejaculate characters, such as sperm length, velocity, even longevity. These can all be under selection known as post-copulatory sexual selection. Now, another component that we are interested in is how about the parasites or endosymbionts that are transferred during mating? Now, these are another organism that plays a role, that could play a role in co-evolution. So what role do these parasites actually play in establishing those barriers that we talked about to reproduction? And that's pretty much what my research group is currently doing at NUS right now. Much of the work, as Eleanor mentioned, it revolves around sexual selection and its role in biological diversification. And we predominantly work on insect fauna, and we have a vested interest, of course, in understanding mechanisms of speciation in Southeast Asia and in Singapore. Now, just for the purposes of today's talk, I'm going to focus on what we are doing with respect to dung beetle diversification in Singapore and the region. So I will navigate through how we use modern DNA sequencing technology to estimate biodiversity in dung beetles. But before I go on into talking about my work, first I want to highlight the students that are involved in this work. And none of this would be possible were it not for these students. They include my grad students, Kai-Sin and Sean, as well as really industrious undergraduates such as Ita and Hilary, who play a major role in generating this data. So if anyone who's ever listened to any one of Eleanor's talks or even read her stuff know how important dung beetles are. So they provide numerous ecosystem services, which include important roles in decomposition, in waste removal, in soil aeration, as well as seed dispersal. And they have been used in numerous studies as indicators of forest disturbance and general forest health. Now, there are about 7,000 dung beetle species worldwide, but the taxonomy and systematics of Southeast Asian fauna in particular is largely unresolved. And as an evolutionary biologist interested in sex, dung beetles are also fascinating because they are strongly sexually dimorphic. And remember when I mentioned where males and females differ in how they look, that's likely an indication of strong sexual selection. And dung beetles are strongly sexually dimorphic. And even closely related species and populations can differ with respect to variation in sexual traits. Now, as I mentioned, in Southeast Asia, there are, I think, about 700 identified species or so. Eleanor can correct me on this, from about 42 genera. But much of the classification is based on esoteric sampling. And as of now, we don't really have a comprehensive checklist, or nor do we have really updated keys for, even in Singapore, a small country like Singapore. And so a lot of ecological surveys thus far clustered unidentified specimens into morphospecies for species estimation. And one potential problem is also that many studies tend to exclude females because females often look similar in external morphology, and it's not very easy to use diagnostic characters to separate them. So there are several challenges, and there are also a potential confounding factor is the presence of cryptic species complex. And I will explain a bit about this later on. So morphological species estimation, as we know, is based on how things look. But there is actually a potential solution to get a more accurate estimation of biodiversity. And that would be to take a complementary molecular approach to biodiversity estimation. So the molecular species estimation is based on a threshold of intraspecific genetic variation. Now, this is just based on a simple principle that individuals that belong to the same species interbreed regularly. As such, they should be more genetically similar to one another than unrelated individuals. So using a molecular threshold, we can characterize individuals that cluster together. Let's say they differ by less than 3% with respect to the molecular barcode, then they are likely to belong to the same species. And so this is actually a very useful tool that goes hand in hand with what we would call as traditional species estimation. So the earliest dung beetle collection records for Singapore were from the 1960s by D.H. Murphy, who recorded about 11 species based on taxonomy and a few other undescribed species. Since then, ecological surveys usually routinely identify only about 5 to 7 species based on taxonomic diagnostic characters, and often about another 5 or 6 that are unidentified. So we've had a couple of surveys over the past few years, and until recently, thanks to Sean, my PhD student's work, we've now have one of the most extensive samplings across the island. So not just within the central catchment and in Pulau Ubin, but throughout the island in all our pockets of forested as well as peri-urban areas. And so when we do dung beetle collection, it's not exactly a glamorous job, as it implies. It involves different kinds of dung. We usually use cattle dung, pig dung, but surprisingly, the dung that works the best is our very own. And I'll let my student explain it.
Common portrayal of dung beetles, right, is that they roll the dung into a ball and then roll it away. So in Singapore, only one species of dung beetle rolls the poop into a ball. The rest of them actually either sit in the shit or they take the poop and they bury it directly where the dung bed is. So as for why some of them will roll it away, right? So the males will try to make a nice dung ball and then use that to attract the female. And sometimes males also, right, they will fight over the dung ball because like they're too lazy to make their own. So they will try to hijack somebody else's dung ball, then they'll fight over it. And then another male will come along and steal it. Because the females will lay her egg inside the dung ball, and the quality of that dung ball will affect the quality of her offspring. So she's very, very choosy. My research focuses on reproductive evolution of dung beetles in Southeast Asia. So looking how a lot of these different sexual features, including the horns, as well as internal features like their ST size, their sperm characteristics, how all of these come into play in terms of how they speciated through Southeast Asia. And once you settle on stuff like what sites you're going to set up, what kind of method that you're going to use to collect them, and first step is to procure your bait. So you have to collect. Squatting toilets are my best friend. Just squat and pull a ziplock bag over my butt, and then straight side. I mean, after a while, you find a lot of different ways to not have to touch the food, like if you roll the edges of the bag. We have an anonymous freezer deposit, right, where people just go in and drop bags of food inside. But to standardize the kind of food, right, to make sure there's no bias, if like somebody's food is more attractive than the other, you have to mix everything.
So never did I realize that my contribution to science would be documented in a box full of human shit labeled Dr. Nalini. So yeah. So imagine now that we've collected all the various dung beetles using different kinds of traps. Next is to establish what they are. So we follow through the typical steps with respect to establishing species diversification, right? We first find the specimens that we have here. We sort them into morphospecies based on high quality imaging as well as diagnostic characters that we gain from the literature, and we cluster them into morphological groups. So groups that we think are the same species based on morphology. Now, this is unfortunately primarily just based on male specimens because a lot of the literature doesn't include information on female morphology. And so this is what we get. So after more than about 20 months of sampling, we actually find, just based on morphology, about 21 species in total. We find one large dung beetle species that's prominent, known as catharsis molasses over here. We find a medium-sized roller species known as Paragymnoplurus. And then we find a lot of these smaller guys, these smaller dung beetles that belong to this genus Onthophagus. And within this genus Onthophagus, we find about, based on morphology, but 19 different morphological species, out of which we can identify 12 to species based on diagnostic taxonomic information. But as I mentioned, this is only based on male specimens and about 1,000 specimens collected over 20 months. Now comes the complementary molecular approach. We take every specimen that we have, right? And we also take a bit of that specimen, let's say an antennal segment or a foreleg segment, and then we extract DNA from them. So extracting DNA from beetles is actually not that straightforward because beetles are highly sclerotized. And sometimes this pigmentation that's in the sclerotized their exoskeleton interferes with DNA extraction protocols. So Sean managed to optimize a protocol that is given a really high success rate for our dung beetle specimens. So after we've figured out how to extract DNA, then we amplify and sequence that, do PCR and amplify the DNA barcode, that region, mitochondrial fragment, that region that gives an identification as to which cluster this specimen might belong to. And so as I mentioned, it's often a threshold-based estimate. So individuals that are less than 3%, like here, they likely belong to the same species, as opposed to individuals that differ larger than 3%, they belong to other species. So the nice thing about this process is that we have doubled our entire sample size with respect to species estimation, because now we can use males as well as females. And so our total data set is about 2,000 individuals. And by doing this complementary molecular approach, we have also increased our species estimation. So now instead of one large catharsis species, we find two catharsis species that are genetically distinct. We still find a single roller species. And we now find, instead of 19 Onthophagus species, we find 22 putative species clusters. And what is interesting is that when we compare our approach with the morphological method and the molecular method, we find that we have actually been overestimating those that are dimorphic, meaning they have strong horns or they differ in male and female. And we've been underestimating species whereby males and females don't look so different. So this is a clear indication whereby using the molecular method actually helps to get a better estimate of species diversification in any given area or in any given study. A species richness, sorry. So let's compare our results with the previous surveys. So we've had all previous surveys find about 12 to 13 species, and we've actually doubled, more than doubled that, but we use human dung because we figured it's a nice mixture. Well, not really nice, but it's a mixture between purely carnivorous versus purely herbivorous or omnivorous dung sources. So what are the benefits of such an integrated approach? Well, by studying variation in both morphology and molecular data, we now have a better estimate of dung beetle species richness. And we can then investigate particular case studies and try to understand the role that certain traits might have for diversification. So we can take images, we measure these species, we measure everything about them from both the reproductive traits as well as traits that have to do with survival. For instance, their wing loading, which is important for dispersal. The overall body size, which is also plays a big role in survival. The exposed eye area, as well as their leg length, and a bunch of whole other traits. And we can then put information about these species on the web. So now we can have a publicly available access to diagnostic morphology for not only individuals studying dung beetles in Singapore, but also those that have representative populations in other parts of Southeast Asia. And the nice thing is that we give representation of both males and females. So one of the main benefits is that we are now able to identify intraspecific polymorphism. Now, for those who are not familiar with what polymorphism is, just look at us, look at your neighbor. For non-biologists, polymorphism basically refers to individuals belonging to the same species and are able to exchange genes, but yet we look different. And so you can think of them as genetically identical, but morphologically distinct individuals. And we've found a couple of examples of this across our data set. For instance, in this species known as Proletarius, the museum records have only shown a 3-horned morphology, morph. But what we've found is that we've actually found there's 3 distinct classes of males within this species. That means they are genetically identical, but they have 3 morphological traits. And there's been no records of them in the museum records thus far. And there's also never been mention of this hornless morph before for this species. And we find such an example again in yet another species known as Orientalis. Again, museum records often have this large morph recorded, but not the intermediates. And we also see that they are statistically different and often associated with overall body size, meaning individuals that are larger have the 3 horns, individuals that are really small have no horns at all. But we also find species whereby there's no clear relationship between horn morphology and body size, meaning there's high intraspecific variation. The museum records often have these 3-horned morph, but we find variation with respect to both the head horns as well as the thoracic horns that is not necessarily correlating with body size. So this is actually a really interesting case study, and that's one of the projects that some students are trying to further investigate. So as an evolutionary biologist, variation is the foundation for differential selection. The moment we see samples that vary, that's when our juices get flowing, you know, that's exciting to us. And so when you have traits that vary, we can then quantify intraspecific variation and calculate selection. We can actually find out what is the intensity of selection acting on different species. So how do we do that? So this is known as allometry. So as I said, for those who are not familiar with this, I'm just going to give a simple explanation. So let's say there are usually traits that vary with body size. Now, if you have body size here on the x-axis and a trait, let's say the length of your hair, length of your arm, any kind of trait, right? That is on the y-axis. And when you take log transform data, if it gives you a slope of 1, that means there is they increase to the same level. But most traits actually have hypoallometry, meaning they are less than 1. So for every unit increase in body size, that trait increases by less than 1. What is interesting is actually hyperallometry, which implies that there is strong differential investment in that particular trait given body size. Meaning for every unit increase in body size, that trait is increasing by more than 1. And so all these sexually selected traits that we see in nature, like the peacock's tail, the antler's horns, and even the sperm of Drosophila, all these sexually selected traits actually have strong allometries of larger than 1. Meaning for every increase in body size, these traits are increasing many, many times more. And that's exactly what we find across all our dung beetle species. If you look at the horn morphologies, they are often positive and larger than 1, and they differ strongly between species. And interestingly, we find that they can also differ between populations within a species, which suggests that within populations, you might have different mechanisms or different intensity of sexual selection acting on them. Now, another big useful benefit of this complementary molecular approach is the identification of cryptic species complexes. So, as contrary to the previous point, cryptic species are basically morphologically identical, but genetically distinct clusters. So there are cryptic species in nearly every metazoan class and phyla, including insects, fishes, mammals, birds, you name it, you can find cryptic species. And there are analysis of genetic data has actually been a powerful tool to reveal such cryptic species complexes in different fauna. And that's so what we found. We found numerous examples of cryptic species groups within our data set, whereby there were initially, let's say, these examples of these two species that were often misidentified or clustered at one. We now find that they're actually genetically separate and similar to these guys and these guys over here. Now, especially for this last group, we might think that, oh, they look very different in body size. But the problem is that the body size is actually a continuous character and their error bars overlap. So it's hard to use just a continuous character as a distinguishing factor to differentiate these species. So what actually drives the formation of such cryptic species? Now, there can be numerous ecological and evolutionary forces at play. Now, if you think about species that look alike but live in different places, now this is known as allometry. If the environment they live in gives them similar challenges, then they can evolve similar morphologies to those challenges independently. So that is a form of what we know as convergent evolution. But you can also have cryptic speciation that happens in sympatry, meaning at the same place. That can happen when individuals of one population look different, look the same as individuals of another species, but they don't mate with each other. There's likely selection on traits that have nothing to do with how they look, but maybe what they eat or what kind of courtship they display. So that's what we are trying to do with this ongoing research program that we have on catharsis molasses. So catharsis molasses is basically Singapore's largest dung beetle of record today. And it was first described by Linnaeus himself. And it's actually been long regarded as a single widespread species across India, China, and Southeast Asia. But when we looked at the genetic information, so we downloaded all the CO1 barcodes, the DNA barcodes from GenBank, and we compared these barcodes across the different specimens, and we find that they are actually quite in line with speciation via allopatry, meaning there are multiple putative species that look similar but are not exchanging genes and are probably largely separated by geography. But interestingly, we also find a case of potential sympatric speciation, meaning individuals within Singapore and Malaysia, they form two separate clusters even though they are found in the same location. And they are genetically distinct. So this gives us a chance to investigate speciation in sympatry. So now just to recap what we talked about very early on in the talk, there are potential barriers to gene flow, right? They can be environment. So these two populations in these two species of dung beetles that exist in Malaysia and in Singapore, they could be separated by geography, maybe dung preferences or seasonality, or they could be separated by traits that are involved in territory, like pre-copulatory, or traits that have to do with sperm characteristics that are post-copulatory. So just to summarize the whole study thus far, what we have thus far, we actually find two kinds of clusters in Singapore and across Singapore and Malaysia. So we find a rare species as well as a common species, something that we find quite easily. But within the rare species, we have individuals that are both from Malaysia as well as Singapore, and similar for the common species. And what is most important is that the specimens of both types are often found are often attracted to the same dung bed and are often found in the same pitfall traps across our sampling. So it is unlikely that there are these environmental factors that are potential barriers to gene flow. So what about reproduction? So that's where we look at allometry once again. That's why I introduced it earlier to you guys. So if the allometric slopes differ between the two types of species, then it could be that the intensity of sexual selection acting in those species might differ. But what we find is that there's actually no difference in overall body size and absolute horn length. And the relative horn length, and meaning the allometry, is of the rarer males, it's slightly higher, but it's not statistically significant. So we then look at post-copulatory traits. So we've been looking at, for instance, differences in the testis size, the genital shape, as well as as well as accessory glands, which are the structures that produce the proteins that are important in ejaculates. Now, what is cool is that we also find differences between the rare and the common clusters with respect to the degree to which their sperm looks, the variation in the sperm morphology. For instance, those in the rare cluster happen to have this hooked sperm head, but those in the common cluster do not have that hooked morphology with respect to their sperm. So part of the ongoing work is trying to understand what is the gene expression in those structures, the structures that are important for the ejaculate as well as the sperm. And that is the work that is being led by our research fellow, Nalini, who is currently taking a comparative transcriptomic approach to understanding these mechanisms of diversification. Now, this part is going to be a bit molecular, so I'm going to try and simplify it as much as I can. And if there's something that you're not familiar with, you can always clarify later. So what we show here basically are a few genes that are highly expressed in the accessory glands, those structures that produce the proteins for the seminal fluid, as well as the testes, the structures that produce the sperm. And what is interesting is that we find that among these highly expressed transcripts, there's actually no overlap between what is produced by the accessory gland and what is produced by the testes, meaning these two reproductive structures have clear functionalities and are both important in producing an ejaculate. And what is cool is that several of these genes are actually novel, meaning they're not shared with any other insect group. And in particular, we see in the accessory glands, there is nearly 50% of the novel genes are specific to catharsis itself, this particular species, meaning that these genes are likely have important functions in reproduction and are rapidly evolving in the genomes of these beetles at the species level. And now novel genes are also, interestingly, some of the most highly expressed ones. So they are candidates for post-copulatory reproductive isolation. And finally, here is a heat map of those top expressed proteins in this accessory glands. And we see like a large proportion of them are functionally important to a novel to catharsis, and they are important, and some are actually novel to dung beetles as a whole. They've been found in other dung beetles, but their functions have not been elucidated. So it's still not that clear. So this is generally work in progress. And what is interesting is that we find a lot of protease inhibitors, heat shock proteins, as well as combination of other genes that are important for protein synthesis. So I'm not going to go too much into detail, but let me just summarize this all by saying that this reiterates the important role of post-copulatory sexual selection in potentially mediating speciation. So now what we have ongoing is that we have a current work. Well, this was interrupted by the CB, but we have specimens that we're planning to send out to do population genomics using both museum samples as well as fresh caught specimens. And we're trying to look at how gene flow has varied across a larger geographic area. And that's for catharsis, of course. And so this is just to summarize. We find that in catharsis, genetically distinct individuals look identical based on external morphology, but there are likely strong post-copulatory mechanisms preventing them from mating and driving speciation. Now I'm going to wrap this off quickly by just giving you a snapshot, a little insight into a very interesting research program that we've just got started. So dung beetles can host a lot of endosymbionts, such as nematodes that are found in the dung. And a recent study in PNAS found that these endosymbionts, these worms, these nematodes that are living within these dung beetles might not be actually parasitic. They might actually be helping to increase the fitness of these individuals. Fitness being those individuals that have more of these nematodes are able to inject them into the dung balls where the young are developing, and they help terraform the dung ball bacterial and fungal community so that it increases larval survival. So that's what we were interested in. We were interested to see, do our dung beetles have these worms? And it's kind of gross, but yeah, they do have tons of these nematodes. And so when you dissect an individual, you'll see them all just coming out. It's quite a interesting experience doing that. So the next question was, do the loads of these worms differ between males? Because in sexual selection, remember I told you variation is the key. If there is variation, that implies there could be selection. And so what we find is that indeed, yes, there is strong variation. Most individuals have few or very little worms, and there are only very few individuals that have a lot of worms. And we also find that this frequency of worms can differ between populations. So this is a potential nuptial gift, meaning the males are transferring these worms to the females during copulations, and the females are using them to terraform the dung ball and increase the survival probability of their offspring. This is potentially a nuptial gift. So that's the ongoing work we have. First off, we want to know whether these worms are specific. Now, because these worms are very hard to identify, that's the usefulness of the molecular method once again. So we are able to do DNA extraction of individual worms that came from different, each beetle, and we look at multiple worms per beetle as well. And so what we find is that sometimes we find worms that are specific to beetles, and other times we find worms that are more generalist, that occur in more than one species as well as even across genera. So as I said, this is still ongoing work, but I just wanted to just share this with you guys because we are quite excited about this. So just to summarize, we find that worm loads can differ within beetles of the same species, and some worms are host specific, others are not so. So after all of that, this is the summary of today's chat about dung beetles. There is a there are clear benefits to using this complementary molecular method in species estimation. And what we find is that the sexual selection on reproductive traits especially differs not only between species that are found in the same area, but also among populations of widespread species. And we've identified that most likely post-copulatory mechanisms that have to do with sperm ejaculate female interactions might be playing a role in cryptic speciation. And finally, that sexually transmitted and maternally inherited worms such as these nematodes might be actually important for fitness, but we're not actually sure if they might play a role in gene flow. So the work that I've just shared with you today is, as I said, very much the efforts of all these students in the lab, and we have a group of students that have been working on this reproductive evolution of dung beetles. But we also have students that are working on sexual selection and natural selection, the trade-off between these two forces in dung flies. I don't seem to be getting away from dung, but or waste in general, because there's another research program that we have, which is a bit more translational, where we are trying to understand the reproduction of black soldier flies in waste management. Because in Singapore, we have a lot of food waste, and we can use these flies to actually convert food waste back into food, basically, by creating compost or animal feed. And we also have a final project where we're trying to look at the endosymbionts, but from a bacterial perspective. Because Singapore has been using Wolbachia as a method of mosquito control, a potential method of mosquito control. And so part of one of my grad students, Hui Ching here, she's leading some students in understanding how these Wolbachia or reproductive bacteria can result in co-evolution with respect to mosquitoes. Yeah, so that's pretty much it. First and foremost, I need to thank all the members of the Repro Lab. This has been our lab meetings these days, all through Zoom, and also the different funding agencies. And thank you guys for your attention, and I'll take any questions you might have.